This one.
The delegates, we are starting our second session today, which is on AHTEG on risk assessment. I would like to invite the chair of ad hoc technical expert group on risk assessment, Ms. Maria Ruohonen-Lerner, to provide a brief background on the group's mandate and to describe the outcomes of the group's work. Ms. Maria Ruohonen-Lerner has a PhD in genetics from the University of Helsinki, Finland, and has also done research work in United States, in Iowa, and the United Kingdom. She has worked at the Finnish Environment Institute since 1995 and at the Ministry of Environment during 2019 to 2020. Maria has participated in the Cartagena work since 2006 and the CBD work since 2014. She has been a member of several risk assessment ad hoc technical expert groups and is the Cartagena national focal point of Finland. Chair, you have the floor.
Thank you. Okay. Okay. Dear colleagues and friends, I'm very excited to be here. It's a great pleasure, of course, for me to present our work to you. And of course, I am in the same time, I'm very honored to have the possibility to present the work of the AHTEG. And of course, I have been very honored to chair that group during the last half a year. And I'm particularly excited about the fact that I have grown about 30 centimeters during lunch, and it must be the good food that I get here. I can explain to you that I got a little extra podium so that you would see me better. I was already introduced to you, but let me say the following. I come from Helsinki, Finland, and I have the fortunate situation to be in the same time zone as Nairobi, Africa. So any silly things I say are not due to jet lag. So let's hope that I will not say too many silly things. As I said, I'm very excited and happy to be here. Let me take you through the road for a decision, or the road for developing guidance on living modified organisms containing engineered gene drives. Already in 2018, the COP-MOP 9 decided that at COP-MOP 10, it will consider whether additional guidance materials are needed for LMOs containing engineered gene drives or for LM fish. Also, you all remember that an important step was taken at that meeting. There is a process for us to use when we are considering further guidance to be developed, and that process is introduced in Annex 1 of the Decision 913. So this was 2018 already. In 2020, just when the COVID pandemic had started, we had an online AHTEG meeting that worked on 2 commission studies, both on fish and the engineered gene drive organisms, and recommended that guidance materials should be developed for LMOs containing engineered gene drives. And then 2022, indeed, COPMOP 10 made a decision that parties made a decision that additional voluntary guidance materials should be developed that should cover general considerations of LMOs containing engineered gene drives and have a focus on mosquitoes. Next slide, please. For this purpose, of course, an AHTEG was established, and a detailed outline for the guidance materials was commissioned and drafted by the ICGEB. The work was started, you remember 2022 COPMOP in Montreal and in snow. In 2023, in April and August, we started the work and had online discussions, first on information that was provided by parties, but also then on the draft detailed outline. Then we had the pleasure to meet for the first time. The AHTEG had the pleasure to meet for the first time in November 2023. And what maybe one of the main things that happened at that meeting, and I will tell you more about that, was that we decided, we had a detailed outline developed for us, which was based on, of course, Annex 3, the risk assessment annex. But at that meeting, the AHTEG decided that we will take a step forward and use something that we call problem formulation approach when developing the guidance. And I will later explain to you how that correlates with Annex 3. So that decision was already done in our first meeting in November 2023. And in order to be able to do the work that we had been given, we set up small drafting groups that worked intensively through November, I think mid-November until early January 2024. Zero draft, a little more developed draft, commenting periods, meetings through online, etc. Then what happened is that I, as a chair and with a great help of the Secretariat, then combined the texts that we had. And that was by the end of January this year. Then final commenting on that combined draft and the meeting document was produced in February this year. And then the second AHTEG met from 27th February to March 1st. Here you can see us. I think this is the first meeting, if I remember correctly. Yes. And then I think there is a picture for you of us meeting at the second time. And then I want to explain a little more for you how we worked. Of course, as you all have gathered out, this was a very short period for the AHTEG to produce something that they have or we have together produced. And what we, as I said already, we decided that there would be 5 groups, drafting groups, that started their work already in November 2023. It was on general issues, now drafting a general guidance on risk assessment based on the problem formulation approach. Then there were, as we called them, they were the mosquito people. They had a group of their own, the group B, Then we had very important Group C that dealt with related issues. I will say a few more words about that later. And then there was a group for monitoring, and then there was a group for list of terms. Each group had a party co-chair that was assisted by one of the experts in that group. Each group had regional representation and so on and so forth. And only the list of terms group was not with a co-chair, or there was just the co-chair who was working on that. I'll say a few more words at the end about this. As such, each group organized themselves. Meetings that they held was always attended by myself and the Secretariat if possible. And of course, all this work was built on existing documents from WHO, OECD, EFSA, NASEM, and scientific literature. So this is how we worked, basically from, as I said, mid-November until mid-February. So quite intensive times. Many of the AHTEG members for sure sacrificed their weekends and holidays and their families to get the work done. So the second meeting we had a combined document, a further refined document with the comments that we received early February. The meeting met in plenary in the second AHTEG meeting. That's how we commonly, of course, work in the AHTEGs. The AHTEGs are not such big groups. We went through the document and finalized the text in plenary, but also in groups. So depending a little on what was needed, maybe on certain topics or certain parts of the document. This is how we worked. And as I have hopefully said many times, work was very intensive. We worked very long days, but the group was very much committed, and that I will like to express my sincerest gratitude for. Next slide, please. So let me then take you through what I could probably call an overview of the document. As you know, the document is long, about 50 pages as a core document, and then of course the different annexes, the references, and the list of terms. But let me try to go through the document so that you get a better grip of what it contains. So the section 1 describes the objective and the scope, and an overview of decision 1010. I tried to already highlight that a little in my maybe opening remarks. Then the section 2 introduces engineered gene drive containing LMOs, explains precautionary approach as an overarching approach, and establishes the context of the document. Section 3 details on engineered gene drive strategies, as well as opportunities and risk concerns. This was, of course, very important for us to look at both aspects of this technology. Next slide, please. Then the section 4 is probably what we should call the core risk assessment part of the document, the rigid risk assessment approach that we have now been taking. And it addresses the steps of problem formulation. I will get back to problem formulation a little later. And it also introduces how testing of hypotheses. I will again go a little more in detail to that later. And in order to test the risk hypotheses, we are, of course, dwelling into sources and quality of information, modeling, comparators. We are also describing tier-based testing, limits of concern, and weight of evidence and uncertainties. So that was section 4. Next slide, please. Section 5 is an important part. You all know that Annex 3 contains advice for us to to provide a recommendation of acceptability of risk and identification of risk management strategies. And there is also a section dealing with this. Section 6 deals with monitoring, and Section 7 describes what we call related issues. And the biographic references are included in Section 8. Next slide, please. I say a few more words about related issues. That's how we decided in the AHTEG to call them. These are the issues that are not per se in the very core of the scientific rigid risk assessment, but very, very important things to consider and look at when you are doing your risk assessment. And the issues that we provide some guidance on are as follows: risk assessment and assessing the benefits as components of the decision-making process. Consideration of benefits to human health, of course, when we are dealing with engineered gene drives, and so this is a particular important part, of course. We are also guiding on how to deal with socio-economic, cultural, and ethical considerations. We have a section on free, prior, and informed consent of IPLCs. Next slide, please. Let me see that I get there myself. Continues what Section 7 contains. We have a subsection on public awareness, education, and participation. Exemplaria gratia, full and effective participation of IPLCs, access to information and risk communication, very important issues to be considered during your risk assessment. There is also a section on comparing novel and alternative strategies to the EGD strategies. There is a subsection on transboundary movements, and there is a short section on consideration of liability and redress elements. Next slide, please. The annexes deal with modeling uncertainties, the WHO guidance framework, the taxonomic classification of Subsidiary, mosquito vectors of diseases, current landscape for the development of IGD LMOs, engineered gene drive systems. And then a list of terms with citations is also one of the annexes. Next slide, please. How did we deal with the mosquitoes? The decision 1010 said that we should look at general issues related to risk assessment of EGD LMOs, but as you remember, we agreed together that a specific focus should be on mosquitoes, while we all know that's where the development is going, or the development is most advanced, I think I dare to say, how to deal with diseases like malaria, Zika, and others. And that's why mosquitoes, of course, that's why we decided that we look at mosquitoes, specifically on mosquitoes. We decided in the AHTEG that mosquito-specific issues are introduced as, we call them boxes. They're quite big boxes sometimes, but they are incorporated into the main document that deals with the risk assessment, the Section 4. Of course, also to Section 6, which is monitoring and other sections. But that's how we decided to approach this issue. And next slide, please. Well, then I go to problem formulation. Problem formulation, the way I would like to describe is a, maybe I dare to say, a refinement of how to approach risk assessment in a focused way. I think the most important things in problem formulation is that we as parties look at what we call protection goals, general protection goals. Biodiversity, ecosystem services, issues like this. broad issues that we want to protect in the environment. Then, of course, we all know that assessing these broad protection goals or issues that we want to protect in our environments would not be possible. And due to that, we are choosing something that we call assessment endpoints. And examples of assessment endpoints would be species, certain species that you choose as to be protected. If it would be ecosystem services, it would probably be pollination and so pollination, or those insects providing the pollination. Or it could be certain type of habitats that you want to protect. So you're choosing, you're narrowing down what you want to protect in order to take a practical approach, and also you need to prioritize and have a clear understanding what to do, what to look at. Then, of course, the next step. So here I've described to you the first and the second step. So protection goals, what do you want as a party or as a region? Second thing, how can you achieve what you want? What species would you pick up as those that will tell you what is happening up in the nature? What type of habitats would you look at? What type of pollinators would you look at? The third important step is that you need to set up something that we call plausible pathways to harm. Basically, this is exposure pathways. Will your stressor, which in this case is the engineered gene drive containing LMO, will it reach what you want to protect? Is there a pathway? Is there an exposure pathway for something to happen, something that you don't want to happen? The next steps, the stream is here to describe the pathways. Anyway, so the step 4 would be then you formulate, of course, risk hypothesis. You look first at the pathways, the exposure. I will have an example for you that was developed by the AHTEG. I have an example for you. So can this happen? What would be the pathway? How would the stressor move to what you want to protect? Then, of course, you formulate a risk hypothesis in order to be able to test and look at the likelihood and the consequences. Then the step 5 is a very important step, and I think I personally think that it's very good that the problem formulation highlights this clearly, that the participation and engagement of stakeholders and indigenous peoples and local communities can be included at all points in the process as appropriate. So any kind of discussions at any point of your risk assessment is useful, is important, is advisable. Next slide, please. So this is I always find it a little intimidating to introduce this kind of tables or paragraphs for you, but I'll do my best. This is what you find in the document, and we the AHTEG found this useful so that you would see how it compares with our Annex 3 of the Cartagena Protocol. So we had above this, you all have been doing this. You have your general protection goals. You have chosen your species that you want to protect, the whatever it would be to narrow down in order to know what to follow. And then, of course, you need to know the novel characteristics of your, in this case, it's an engineered gene drive containing LMO. And then you move to pathways to harm, and as you can see, they are different steps of pathways. The next slide will show you a little more, and each of the step has a risk hypothesis. Will this happen or will this not happen? Of course, and then you need to test it. You have different methods to test your step. The next one will explain a little more. And then after having done this, then you, of course, do an overall risk evaluation, and then you do your recommendation for a decision. So I hope I've managed to explain that the problem of formulation approach does not in any way deviate on what we have been doing and what you all have been doing, but now there is a more rigid way of explaining how to prepare yourself for risk assessment. What do you really want? What can you do? And what kind of methods would you have? And of course, especially the exposure pathways are very important. Will this happen? Will this take place? If it happens, what do we do? And I think, of course, at the end of any risk assessment, as we know from Cartagena, we prepare a recommendation. Of course, it's very important in that recommendation to consider all the information that you have, look at possible uncertainties, etc., etc. And then, of course, risk management as always. Then I have my final slide. How am I doing time-wise? I'm sorry? Okay. Anyway, I will anyway show this last one, whatever I'm doing time-wise. But it will be my last slide, and then I'm happy to, of course, answer any questions, and I'm in your hands. So this is a pathway that the AHTEG came up with, and the risk hypothesis here is that the consumption of EGD LMM living modified mosquito adults by insectivorous bats has no greater toxicological effects than from feeding of non-EGD LMM adults. So basically, we are asking ourselves, will something happen to the bats if they eat EGD LM mosquitoes? And then we start to look at the exposure pathway, which here is, first of all, intentional release of the mosquitoes. These mosquitoes mate with target mosquito populations. The frequency of genetic modifications increase in target mosquito populations. The bats will eat these LM mosquitoes, and this increased feeding of modified mosquitoes has a toxicological effect on the bats. An abundance of these bats is significantly reduced. And of course, at the end, because the bats and many of different organisms in the environment assist us in pollination, then we also the harm that we would then see, of course, decrease in a population of the bats, if that is what we want to protect, and then of course also a possible decrease in ecosystem services. So that would be the pathway. Our risk hypothesis is, of course, it starts by that nothing happens, and then testing each step in the pathway, as is shown on the right, is how we then move forward. Each step is tested. Of course, if this step cannot, or this step will not happen, then you don't have to continue. Of course, when you look at each step, you always also consider the uncertainty that you would have analyzing those steps. And I think I'm done with the presentation, but I would like to thank well, most of you, of course. First of all, you all being here and listening and showing interest into this work. I would also like to take the opportunity to of course to thank the Secretariat. especially the dream team biosafety. Of course, the chair of the Substa. Many thanks. But in particular, of course, all my colleagues in the AHTEG, because without you, this would never have happened. And I have written down the names of those of you who are here, so you'll get my special attention. So my many thanks, heartfelt thanks go to Lilian from Malawi, to Maturin from Burkina Faso, to Kamal from IPLCs, to Ricarda from Federation of German Scientists, to Eva from Third World Network, for Brinda, Foundation for the National Institute of Health, and for Felicity Global Industry Coalition. So many thanks, and thank you for your attention. Okay, I was asked to continue. Any questions that I can help you with? Yes, the gentleman over there.
Thank you very much. This is not a very much holistic question, just a small comment on the last illustrative pathway. I'm being a biologist or ecologist, a point comes to my mind that although it's an example or the illustrative pathway, but the impact could be positive also, or not on the negative on the bats. It could be positive in the sense that the population can be increased. Impact can be any type of impact. So this is just a very small point that came to my mind. Thank you.
Yes, thank you very much for that comment. Indeed, of course, this could be the case, and of course, as we are dealing with this, that how to look at both sides when we are doing our risk assessment in the related issues. There is a topic, but thank you very much for that comment. Yes, please.
First of all, thank you very much for this very interesting presentation. On the issue of related issues, I was very pleased to see something about ethical consideration, because I've been trying to raise the issue of ethics in biodiversity, but it looks like not yet well understood, although they are very, very important. Secondly, on the issue of mosquitoes, all of the research have been done, all the tests have been done. Our colleagues from Africa, they still think that malaria is still a real problem, meaning that there is some confusion about the whole aspects of gene drives regarding the mosquitoes. Thank you.
Apologies, I couldn't hear you very well during your second. First of course, thank you very much for your kind words. Apologies that I didn't catch
This one
your second.
is related to ethical.
Yes, this I heard, sir, but the second
the
one.
Second one,
yeah, please.
the outcomes of all what already have been done on mosquitoes are not really very encouraging based on all of the reviews that we read from time to time. Just I need clarification for that as well. Thank you.
Okay, thank you very much. Anything else that I can help you with? Yes, please, Osama.
Yeah, just related to that was the issue of null hypothesis taken on board in the evaluation. I mean, would the mosquito behave in the way that they are expected to behave? Because I mean, all the reports that has been there have shown that it has very little effect on affecting really the malaria. So was the issue of null hypothesis bear in mind not just the issue of what could go wrong related to the new introduced trait, but the null hypothesis, will it behave as designed or as expected? Thank you.
Again, apologies. I was not in the malaria group, sorry, the mosquito group. But indeed, of course, malaria was discussed and other diseases as well. But here, I think our aim was to to provide guidance how to assess the possible impacts, and there is a section on of course on the biology of the mosquitoes, and I think it reflects also the issues that everything doesn't go always as planned and as the development would have hoped to go. Let's put it that way. So yes, this is, I think this is considered both in the document and of course was discussed at the meeting. Thank you. Anything else? Yes, please go ahead.
Thanks so much, Christopher from Zambia. I am glad this document seems to speak to our minds, because the way the assessment has been drafted and done appears to be addressing the gaps of assessment in such a way that it will address on average, or if not everything, on the unknowns that may occur, that may appear to disrupt research that may need to take place. So I am happy with the outcome of the AHTEG. Thank you.
Thank you very much. Let me thank you on behalf, of course, of the whole AHTEG. Thank you very much. Yes, please go ahead.
Yes, Musa Tabadogo from Burkina Faso. Just to add my voice to thank very much the AHTEG and to congratulate the whole team of AHTEG for this outcome. You have done a wonderful work for Africa. Africa is, malaria is an African problem. And here you are giving means for innovations. So this is very important. Yes, we focus on risk, and even the title of the guidelines is risk assessment. When are we going to talk about the benefits, the risk analysis and benefit analysis? When are we going to talk about that? Is there any possibility or somewhere where we can see the benefits? Yes, the analysis of benefits. Thank you.
Thank you very much for that comment. And indeed, I think I don't only think, I know that the approach that we took in the group is that we looked at different assessments partly apart, so that the kind of rigid risk assessment is done as focusing on possible adverse effects. However, there is what we now call in the document the related issues. There is both guidance on how to deal with or how to reflect both the risk assessment and benefit analysis in decision making. That's one issue where there is guidance on, and then there is also an additional subsection on benefits for human health, exactly because of the reasons that you raised. So I hope that is helpful. Thanks. Anything else I can? Yes, Lillian, please go ahead.
Thank you, Chair, and thank you for that presentation. Just want to add in terms of the benefits. The document has spelled out some benefits that we are there in terms of the use of gene drive mosquitoes, not only for the health benefit. We have also looked at other benefits, that opportunities that gene drive organisms, living organisms, can also provide. Issues of addressing threatened species, addressing invasive alien species. Those are some of the benefits that have been highlighted in the document. And just also to add, to say that when you look at the whole document, if you go through the whole document, you will see that it can also act as a tool for capacity building. We are aware that not all of us are at the same level in terms of the capacity. So the document itself, from when you look at the details about gene drive, it is also something that one can be able to build capacity on gene drive. And also in terms of the risk assessment, much as we know that this is the principles are the same when we're conducting risk assessment, but when it comes to monitoring, it's very clear that some of the uncertainties that we are anticipating, or maybe we can think about, can be addressed through the monitoring. Thank you, Chair.
Thank you very much for those important additions. Thank you very much. Much appreciated. Yes, Ricardo, please.
Thank you, Madam Chair. Yes, having been a member of the AHTEG, it's right. It just was a lot of hard work, and it was very short the time we had indeed. And I find that we really could have done with more time and more space, because if you look at it, we did not fill out the general part actually with gene drive specifics. There is a lack also in terms of what to do in terms of cutoff criteria. When do we know we actually have too little data? And what do we do with actually the fact that we can't do the experiments? Because once we release it, it's out there. So therefore we rely on modeling, but that will rely on other data, which will sort of not be able to imitate what gene drives will do, and how to deal with that over space and time, and predict, for other receiving environments. And I feel that it has been a real good start, what we've done here, but that we also still should not see this as it will give the answers of how to do the risk assessment, and that now we can do risk assessment because there's so many gaps still in knowledge and how we should deal with them. So I feel we still need to apply the precautionary approach very strongly here. And it will require more work of all of us to figure out, like, where are really the challenges and how can we deal with them? And in terms of, for example, the issue of protection goals and to operationalize them, one really needs to know one's environment so well, and how could interactions, indirect interactions and consequences play out, to have the hypothesis that Maya said that we would need to assess. We might not actually have a clue what might be consequences, and that will then not be available for us to assessing. So I feel we are not there with a product that actually will give us the capacity for risk assessment, but that is a matter of the topic itself, because it's a new technology, and we cannot do the experiments, and because once we release it, it's out there. And I think we need to think further on this and how to deal with this. But it was very hard work, and I think this topic will be accompanying us for quite some time. Thank you.
Okay, thank you for for those comments. Indeed, we have of course raised the issue of precautionary approach, which is a overarching approach that we use in the Cartagena Protocol, and we have also added a lot of information related to how to do modeling and how to assess uncertainties. So indeed, we all have, of course, seen how that this topic is a challenging topic, and we need to look at it from all possible angles. Anything? Yes. Okay. Helmut, please.
Thank you. Thank you very much, Madam Chair. Thank you, Maria. And I would like to thank you very much for a very clear and succinct and structured presentation of what you have achieved in the AHTEG over the last couple of months. In fact, I would like to thank for the possibility of this, how to call it, warming up session before we go to Substa, to get information on important elements which we are going to discuss further throughout this week in Substa. And the presentation of what has been achieved and information to all of us, I think, is a very important pre-requirement that we can informed discuss these issues. I would like to thank you and the AHTEG for the work which you have done and what you actually have achieved. From what I've heard, I think you have explored a very well-functioning structure with your subgroups and plenary on how to develop this huge task in a rather short period of time. And I'm also quite inspired with the problem formulation element, protection goals, assessment endpoints, and how this then relates and fuels into a structured and tiered risk assessment process. So I think you have developed something very remarkable, but without prejudging, of course, our discussions later this week and what we will do with this product. I would like to ask you, because we have heard now by some colleagues some questions and reflections, that there may still be quite some open questions in the one or the other direction. And this is a dynamic field with a lot of developments. Have you discussed, and what would be the answer of your colleagues and yourself in the AHTEG, how to accommodate this in, so to say, the near and also more distant future, how to elaborate further, how to use and develop such a document potentially further according to any progress which we do in science and also in other areas? Thank you.
Thank you for that question. I think we were maybe too busy to very much think about what happens after 1st of March, because that was our deadline in the meeting. But it indeed came up at the meeting that we are all aware of the fact that the technology develops very fast. The possible applications that we will see will develop fast, and we need to have a way to look at this. And we know, for instance, that the WHO guidance on engineered gene drives is something that is regularly updated based on developments in science and based on the experiences that the users of the guidance get. So I think nowadays, when we live in a world where updating documents online is not a difficult task, and so on and so forth. So my personal answer would be that indeed there is always it will not be difficult to update a document like this. I however have to say that I already said that we dealt with the topic of uncertainty exceptionally focused on that discussion. Likewise on modeling, because we thought that these issues will assist us in the world where we don't know everything, which is of course always the case in the nature. So all those issues have been discussed in the group, and I hope that they are reflected in the document. But if anyone else from the group would like to, who is present here, would like to say something about related to this, how you would see the document, the draft document to go forward, I am happy to invite you to say something. Thanks for that comment. On maybe yes, please. I saw another hand. So apologies if I missed someone. Please go ahead.
My name is Barbara Tabrechi. I come from Uganda, and I would like to first of all, thank you very much for this presentation and the work you've done as AHTEG on developing guidance on risk assessment. My question relates to the concerns regarding gene drives. Gene drives are designed to spread and persist in the environment. The example you presented here is generally towards standard GMO crop and not gene drive. Can you explain why you chose this example? Thank you.
You mean the example, the pathway example?
Yes.
Yes, I see your point, and indeed, this was something that we decided to take as an example. Of course, like somebody said earlier here, that of course the basic structure of risk assessment doesn't change if you are using the engineered gene drive systems. So the basic structure you do risk assessment doesn't change, but of course the knowledge that you need changes, and maybe yes, the knowledge that you need will change, the methods will change, and so on and so forth. We found this example that I presented to you as a simple example to be added into the guidance. Of course, fully understanding what you are saying, that this doesn't necessarily have to be an engineered gene drive example. Let me still reflect a little on this the comments on how complicated issue we are dealing with, and we have been trying to reflect that carefully in the document in many ways, and in addition to that, we have also carefully looked at, as I said, uncertainties. The precautionary approach is a overarching approach that we always use in the Cartagena Protocol, but we have also tried to look into the task that we are asked to do after the risk assessment, which is the recommendation. And the recommendation, of course, reflects your level of knowledge, and it also reflects possible uncertainties. So there is guidance how to deal with these issues. That went a little broader than the question originally was, but I hope that I managed to explain a little the purpose of providing quite a simple pathway for you to see. Or we decided to produce one, and which I am presenting here. Anything else I could? Yes, please, Lilian, you have the floor.
Thank you. Thank you. Maybe just to add, I don't know the question, the way it was coming, it seems she noted that it's not the example that we have is not for engineered gene drive, the way she brought up the question, but this was just a simple pathway, but it's for engineered gene drive. And also just to note that there are strategies, two main strategies for engineered gene drive. You have the self-sustaining and self-limiting. So depending on the strategy, you can have different pathways. And this is just an example. There can be several pathways to harm that you can work on. Just wanted to add on that. Thank you.
Yes, that was thanks again for adding these important things. Indeed, what I probably forgot to say about the pathway is that, of course, this is a very simple one pathway example. Of course, the nature doesn't work like this. We would need several pathways. The pathways would have branches and so on and so forth. If you think about exposure as such, from a stressor to the environment. But of course, the way to approach this is to try to have pathways that you can follow and that you can test. And of course, it means that you would need to have more pathways to look at. But as an example how to structure one, this was chosen, and what I think most important was that it was drafted in the AHTEG meeting, and based on a problem formulation approach that we chose to follow in November, and in February we were ready to structure a pathway. So I hope that convinces you that this is something that we all can do to structure pathways. But as was said by Lillian, this is, of course, you will not work on one pathway. You will work on several ones. Yes, please go ahead. Yes, please, Madam.
Thank you. I'm from EDC Group in Mexico, and I really don't understand. Oh, sorry. I really don't understand your answer in relation to this about that is the same structure for risk assessment, because there is nothing like Chindraf's organism in the environment. They have never been there forever. And it's not only novel, it's also that it's designed to persist in the environment, and that is very farther from what you can do in a lab or in theory or in modeling, because it has a lot of new complexities of the interactions in the environment. So it's not clear for me when you explain this work, if you have only worked on the same structure of risk assessment we have today, it seems very insufficient, and it should be followed by a recommendation on not any kind of release until this is cleared, right? Thank you.
Yes, apologies if I have not been clear in what I said, but what I tried to say, I wanted to say that any theoretical risk assessment, when you have a stressor in the nature, follows the same path. You look at the stressor, you look at what could happen, you look at the likelihood, and by looking at what could happen is that you think of the consequences. First, you look at what is there, what can it affect, what would be the effects, and what would be the likelihood for that. And that will not change, whether it's something in the future of a development of genetic developments or any other thing. It's the same for chemicals, it's the same for invasive alien species, that the approach is the same. Risk assessment, this is how risk assessment is carried out, step by step, like our Annex 3 advises us. Now we are dealing with a very complex situation. We are dealing with, in our example, we are dealing with mosquitoes that of course are moving organisms. They are not only semi-natural, they are natural organisms. And that, of course, puts a lot of weight on what we need to know, what we need to look at, and what we need to test. But the structure of the risk assessment will not change. But the challenges are, of course, in us to doing our best in the assessments and doing our best in gathering information, providing further information, doing additional research, whatever is needed for this. And then, of course, I want to highlight once more that the way the guidance is built is to advise you how to do or guide you, of course, on a voluntary basis, like it always is, how to do your risk assessment. But in the recommendations, you then reflect what type of results you got from your risk assessment. And of course, if you have, as a party or as a region, too many concerns about uncertainties, that will be reflected in your recommendations and in your decision making. But the risk assessment has to follow, the benefit analysis as well, has to follow a scientific approach how to do these things. And then you move towards, and of course you're reflecting on what we call related issues. But then you move to recommendations, and that's how you proceed. And this is how we are trying to to structure, or we have tried to structure the document. So I hope this was helpful. Anything else you would like to still? Yes, please go ahead.
Thank you for describing this very complex process. In terms of the inclusion, engagement of Indigenous peoples in this area, including Indigenous knowledge and the concerns that are very much along ethical lines with people who do live and depend on biodiversity very closely. How much of the elements of these would include Indigenous engagement along, whether it's on this risk hypothesis, but also on ethical engagement in this process? Is it included along the line of inquiry into this as well to get information? Because I do believe that this one piece here is a part of a larger set of other pieces that need to be looked at, including when it comes to governments and states and policy and regulation on this particular subject matter. Thank you.
Thank you for that question and comment. Indeed, a very valid and important point, and what we have tried to describe in the document. First of all, saying that, as you correctly mentioned, that we see a very important step in the risk assessment is the involvement of stakeholders and right holders like Indigenous peoples and local communities. This would of course go for an assessment of benefits likewise. In addition to that, I would like to say that in what we call related issues, we have included the Indigenous peoples and local communities in several of those issues. One of them, and there is a separate subsection on free, prior, informed consent. So indeed, this was we had a representative from the IPLCs, and with the assistance of his knowledge, we have included these issues into the guidance, and I hope we have been successful in that. But thank you for your comment. Anything else I can help you with? Any further questions? It doesn't seem to be the case. I'm of course available if you have additional questions during coffee breaks or during this meeting. Let me consult so I know how I'm supposed to behave now. I am released from this position here, and I thank you very much for your attention, and I'm available. Thank you.
But I don't think, Chair, you are released because we need to thank you for all your work done chairing AHTEG. Really, beside you, I wish to thank delegates here who recognized your work. And it is really very good discussion. Just it's supposed to be in this workshop. Thank you. Thank all members of AHTEG and thank the delegates. Now I would like to stop this session, and by our timetable we now go for a coffee break. It will last half an hour, and we will be back in 10 to 3. 10 to 3, yeah. So 2:50. 10 to 3, 2:50. Enjoy your coffee.
please come in and take your seats. The most exciting session is about to start, so you need to be in your seats so you can take part in this exciting discussion.
Dear delegates, we are starting our final session today, which is on AHTEG on synthetic biology and the results of this group. I would like to invite the co-chairs of the multidisciplinary AHTEG on synthetic biology, Ms. Kishma Primus Ormond and Mr. Florian Rabitz, to provide a brief background on of the group's mandate and to describe the outcomes of the group's work. Mr. Florian Rabitz is a professor of political science at Vilnius University, working on environmental policy and novel technologies. Ms. Kishma Primus Ormond is the senior plant protection officer in the Ministry of Foreign Affairs, Agriculture, Trade, and Barbuda Affairs. She works with the government of Antigua and Barbuda through the Environment Division as a leading expert in matters pertaining to biosafety and other related topics. She is also a plant protection expert and have worked on numerous projects and initiatives in Antigua and Barbuda and through the region. Co-chairs, you have the floor.
Thank you very much. And thank you everybody for participating in this workshop. In the next 45 minutes or so, we'll walk you through the main elements of the process as well as the main outcomes. Could you move to the first slide? And we'll keep our remarks fairly general. In case you want to know more details, then we kindly refer you to the documents that are available on the website. We'll start with a bit of general background and introduction. Then we'll move to the methodology that we have developed, the specific outcomes. Then we will discuss briefly the various capacity building, technology transfer, and knowledge sharing needs that we have identified. Next. And then we will move to the review process and the recommendations. Next slide, please. So first of all, synthetic biology is not an entirely new issue under the conventional biodiversity, as you all know. So this has been an issue that we have been addressing here for more than 10 years. There is a variety of previous COP decisions related to synthetic biology. There have been several previous AHTEGs on synthetic biology, although this AHTEG was the first one that was explicitly multidisciplinary in character and that explicitly aimed to develop a horizon scanning process for identifying new trends and issues in SynBio relevant to the convention and its objectives. And at the same time, also, there are parallel processes going on in other international forums. The World Health Organization has been looking to some aspects of synthetic biology that perhaps partially overlap with the process under the CBD. The same applies to the IUCN. And even inside of the CBD, we have processes where there are some overlaps, notably in regards to risk assessment, as well as DSI, where we have throughout this entire process also tried to ensure a coordinated and non-duplicative approach. Next, please. Now, at the core of our work is the operational definition of SynBio that you can see here on the screen. And that definition is, as you will know, fairly broad. It encompasses a very wide range of different types of technological developments in the life sciences. And this has informed the process that we have been implementing over the past year. And this also means that we are dealing with a variety of applications that differ in terms of their technological readiness. We have been dealing with some applications that are in early stages of commercialization, but also on the other end, applications that at this point are mostly speculative in character. And in line with our terms of reference, the purpose was to assess the potential positive as well as potential negative implications of these types of transit issues in regards to the three objectives of the convention. Next slide, please. Okay, so first of all, you see here the overview of the timeline. And to start with, as I mentioned, this entire process is the first time that we are using a horizon scanning mechanism of a multidisciplinary nature for identifying relevant transit issues in SynBio for the convention. And we have been trying to put all of this together in a very short period of time, about 6 or 7 months. We had our first meeting in Montreal in July 2023, and we adopted our final report in January 2024. So that did not leave a lot of time, but I think at this stage, I should also add that in spite of these time constraints, we did, I believe, do a good job, and also despite the diversity of views that sometimes existed in the group, we managed all of this in a very collegial and constructive manner. So in terms of, in line with our terms of reference, the objective of this group was to develop and test a process for broad and regular horizon scanning, monitoring and assessment of the most recent technological developments in synthetic biology. And this process consists of 4 steps. We have information gathering, which is mainly in the hands of the Secretariat. We have compilation, organization, and synthesis of information, which is also in the hands of the Secretariat. And then we have the assessment step, and finally, the reporting step. And I should note that these 4 steps are not necessarily sequential. So in the overall process I will show you in a minute, we can go back and forth between those various elements. And so what we did was that we tried to identify trends and issues in SynBio relevant to all 3 objectives of the convention, and based on that, develop recommendations for further consideration by the COP as well as potentially the MOPS. Next, please. Here you can see the outline of the overall process. So at the very top, that is the very first information gathering step, where the Secretariat developed a list, an initially very long list of trends and issues in synthetic biology based on the outcomes of the previous AHTEG, based on the technical series publication on synthetic biology, and based on submissions received from parties, non-parties, and organizations. Then the part marked in, it's not marked in red, but it should be marked in red. Below that, you find the process itself. Okay, so what we did is we, in the group, narrowed this initial list down to a list of initially 17 items, which we then addressed for further in-depth assessment. Next slide. Yes. So from the outset, what we did is that we had a virtual submission process where each member of the group was asked to select items from an initial selection list or to suggest additional items themselves. So each expert was asked to submit a maximum of 5 items. At this point, we did not differentiate by the number of mentions, so we just used this to compile a first list of items based on the initial list developed by the Secretariat. And so after this first step, we had 33 items from the initial selection list plus 4 new items. Next, please. After that, we had a virtual meeting in October 2023, where on the basis of these 37 items, we went through an exercise of indicative preferencing, where we use a methodology that is known as Z-scoring, where each member of the group assigned a score between 0 and 1,000 to each of the items on the list. And what we then did was that we calculated the standard deviations for each of those items in order to come up with some aggregate measurement of which items are considered most important by the members of the group. If you want to know about the details of that scoring, then you can find that also in the meeting documents. So we then removed all of the items that had a Z-score of 0 or a negative score, and retained only those 17 items with a positive Z-score. So from this list of 17, we again split it into 2, and we developed a list of 5 priority items destined for in-depth assessment, and a list of 12 items for less detailed assessment. And these 5 items, as you can see here, include self-spreading vaccines for wildlife, self-limiting insect systems, engineered gene drives, the combination of AI, machine learning, and synbio, as well as a more conceptual element, the question of inequity in the participation of developing countries in the context of synthetic biology. So those 5 items we assessed in depth. The 12 remaining items we did not assess because we simply did not have time for it. And yes, so to move on, next one. What's the next one? This is the next one. Okay, good. And yes, so thank you very much. And I would now move over to Kishma to present the 5 priority items. Thank you.
Thank you, Florian. I would now like to give the overview of the work done thank you. The work done in our 3rd meeting in January. I will now go in to explain the prioritized items for detailed assessment. You have AI and machine learning. Rapid advances in AI and ML enable novel uses of synbio. Developments include LMMs for risk assessment, biodesign, tools for protein engineering, and cyber physical system with interaction between AI and symbio components, rapid technology process and commercialization, potential to enhance conservation efforts and improve efficiency of resource uses. I will not go in detail. You can find everything else in the meeting document. Next slide. We'll now move on to self-spreading vaccines for wildlife. This is aimed at addressing challenges in wildlife conservation and animal disease management and preventing zoonotic disease spillover. Research in early stages, applications against Lassa fever, Ebola, and rabies are currently being explored, could be used to control the control of invasive species, management of pesticide resistance, and improve disease management and resilience in wildlife. Right. We'll now move on to self-limiting insect systems. Self-limiting insect systems use genetic engineering for suspension of insect population to sterile techniques, technology in early stages of commercialization, potential tools for managing pests as well as disease vectors, and potential for transboundary movement. Slide. Now we'll move on to the engineered gene drives. Engineered gene drives are genetic systems that are transmitted to progeny at super Mendelian frequencies, could allow effective biological control of pests and disease vectors via population suspension or population replacement. Engineered gene, sorry. We'll now move on to inequity in the participation of developing countries. Developed countries lead in SynBio. The need for capacity building in developing countries, research, development, assessment, monitoring, and management of SynBio. The need to ensure access to potential benefits. The need for appropriate regulatory capacities for monitoring and management biosafety aspects of SynBio. Here we already spoke about the items that we assess with a more detailed manner, and we also have additional trends and issues prioritized for assessment. For example, you have the engineered bacteria for nitrogen fixation and fertilizers, and all of this would be in the meeting document. Here we move on to capacity building, technology transfer, and knowledge sharing. Options were developed for independently for other processes. There are 11 options for capacity building. For example, we had building capacity by sharing the results of broad and horizon scanning, monitoring, and assessment, especially for indigenous people, local communities, women, youth, and holders of other knowledge systems. We have 11 options for technology transfer. For example, we have taken into account the results of the broad and regular horizon scan monitoring and assessment is the prioritization of technological transfer. And we had 13 options for knowledge sharing. For example, making use of the knowledge derived from the broad and regular horizon scanning, monitoring and assessment, respecting and understanding the different systems, knowledge, and including its contextual in languages other than English. Also supporting the development of bioeconomies in developing countries could contribute to the objectives of the convention. We now move on to the review of the process. We had general reflections from the group, and all 4 steps of decision 1531 were addressed. We have completed the task in a very short time of 6 months. Sound multidisciplinary process under the convention was developed and provided a unique experience. Additional resources were required by the Secretariat, and we had challenges related to the visa and resourcing. Next slide. So we'll continue to review the process, and we have elements that we found could be useful in improving the process. For the information gathering, we will now, it will be in 2 parts. A1, as you can see on the screen, Horizon Scan, and A2 to inform the process. And we can also submission of information, online forum, literature review, engineering with diverse stakeholders, example, IPLC, women's youth, scientists, practitioners, including other languages. And all of this will be in more detail in the report document. And we'll also move on to the information gathering step, which will also be into 2 parts with the horizon scanning and the information to the assessment step. Submissions of the information, online forum, literature review. We have engagement, sorry, I was on the wrong slide. So yes, elements for improving the process, sorry. So we have B, which is a compilation, organization, synthesis of information, which is 2 phases. As you can see, the B1 and B2, we have peer review is important for literature review, timely and effective compilation, synthesizing information be aligned with to provide the basis of the assessment. And we have seed assessment step, 2 phases. You have the screening and the assessment of the prioritized items. And you can say the screening order, exchange among experts, initial Z-scoring, which was mentioned before by my colleague. And we also have inclusive partnership of experts from different principals, IPLCs, academics, specialized experts. Next slide. And here we are at the recommendations for multidisciplinary experts, for a multidisciplinary expert group, noting that SBSTTA will perform a review of the MATEG, provided options for consideration depending on how its review takes shape. We have 26 potential recommendations on general consideration, artificial intelligence and machine learning, self-spreading vaccines for wildlife, engineered gene drive, inequity participation of developing countries, self-limiting vaccines. And for final consideration, this was the first horizon scan under the convention. Important lessons were learned during the first cycle. An inclusive multidisciplinary process is key, especially for assessment. 5 key areas of synthetic biology were assessed. 12 additional areas were prioritized. 35 different options for capacity building, technology transfer, and knowledge sharing were highlighted. Several different options for recommendations are available to support SBSTTA in their review of the process. Thank you. And as I said before, more details can be found in the report document. I'll now hand over to the Secretariat. Oh, to
Good afternoon, everyone. Thank you for joining us here today. Thank you for the co-chairs for the lovely presentation before us. I'd also like to thank everybody online. So good time of day to wherever you're joining us from. So my name's Austin McLaughlin. I'm a program management officer at the Secretariat, and I will be presenting on the literature review of the trends and issues in synthetic biology. So this is the other complementary process to the multidisciplinary expert-driven process. Next slide, thank you. So just as an overview, the literature review was prepared by the Secretariat with the support of a consultant who unfortunately couldn't join us today, and the financial support of the European Union. There was the first draft of the literature review was made available in January 2024, and then underwent a peer review process into mid-February 2024, and then it was revised for the SBSTTA meeting. This was based on the trends and issues in synthetic biology on the provisional selection list, as we just heard from the co-chairs, which, just to repeat, based on the operational definition of synthetic biology and covered a time period of 2012 to 2023. Really, the aims of this literature review were to be a complementary quantitative analysis for the broad and regular horizon scanning, monitoring, and assessment process. I'll overview some of the details, but there's a lot more to unpack in the actual document itself, so I encourage you to go look at information document 5 for the SBSTTA meeting. So for the methodology itself, the analysis was divided into 2. First, to look at published literature, and then to do a light patent analysis. So for the published literature, this was based on using Boolean search terms in the Scopus database. So we first defined a bucket term to sort of restrict the results within Scopus, and then develop specific search strings for each of the individual trends and issues in synthetic biology. Following this, there was some data cleaning that was done, so further adjustments to the various search strings. And then for some of the trends and issues in synthetic biology, there were a few subsets to try to define and try to capture as many articles as possible. So when summing those, some duplications had to be removed, as well as when looking at sort of overall pictures within the published literature, duplicates would have to be removed to ensure that the quantitative values were accurate. Following this, then the consultant worked on looking at temporal indicators, geographic information, as well as looking at co-authorship collaboration. For the patent analysis, this was done on the patent scope database. So it followed an approach of using the same bucket term, the Boolean search string, as well as looking at specific classes related to synthetic biology. Then, because of Patent Scope's lovely tools, we were able to get various different types of data that could be extracted, such as class and subgroup classifications, as well as geographical information. And then for the analyses, looked at temporal indicators, class and subgroup analyses, as well as geographic information. Next slide. So for the published literature, if you were to sum all of the articles within the 55 trends and issues in synthetic biology, there were just over 60,000 publications that were retrieved. In terms of the top 5 that made up the most publications, these included medical and therapeutic synthetic biology applications, followed by genome-edited plants, followed by synthetic biology-enabled production of antibiotics, natural products, and medically relevant compounds, followed by increased sophistication and expansion of genome editing tools. The 5th being synthetic biology-enabled production of petrochemical precursors and industrial chemicals, followed by advances in protein engineering, followed by integration of artificial intelligence and machine learning. 8th being genome-edited animals. 9th being synthetic biology applications for bioremediation, biodegradation, or biomining. And the 10th being biosensor, sensory devices, and diagnostics. So overall, we could see for the annual publication rate, as you can see on the graph on the screen, there was an increase from in 2012 of about 2,500 publications to in 2023 having 7,900 publications being published. In general, you can see that it's a relatively linear trend, increasing each year. And then for the individual topics in synthetic biology, the trends and issues, there were sort of 2 different types of growth seen. Either it was constant, so the publication rates remain relatively constant, or it increased linearly. In terms of looking at the various trends and issues, I encourage you to look at the annex in particular of the document. You can see that the strongest growth was for those related to genome editing. So these were the genome-edited animals and plants and the genome editing tools. However, there was others that did have other strong linear growth. So this included the artificial intelligence and machine learning, next-generation sequencing and bioinformatics, and non-medical microbiome engineering. So in terms of those that were relatively constant, we saw relatively constant rates of publications in protein engineering, as well as synthetic biology-enabled production of petrochemical precursors and industrial chemicals. When looking at the geographic breakdown within the trends and issues in synthetic biology, we could see that the top 5 countries that contributed, the geographic analysis was based on the lead author's location. You can see that the United States of America, China, the United Kingdom of Great Britain and Northern Ireland, India and Germany had the highest publication output. And then to further look at the collaboration between research groups based in these top countries and their collaborators, we did an analysis where we extracted information from Scopus. And you can see that in the green map, the United States of America and China in the orange map, you can see overall that there are collaborations going on between research groups based in those countries. We saw that for all of the countries that we looked at. And for these top 5 countries, there is a collaboration with at least 100 other research groups based in other countries. So you can see for the map for the United States of America, you can see that the darker colors represent higher numbers of collaboration. Similarly for the graph for China. Next, for the patent analysis, we started by first exploring that overall bucket term that we defined in the literature portion of the analysis. So when we were searching this on the Scopus database, we pulled just over 51,000 patents for the period 2012 to 2023. And you can see on the screen in the top graph, you can see that it is roughly linear, increasing from about just under 1,500 to just over 7,300 from 2012 to 2023. When looking then to see what makes up the potential growth and what makes up the patents within this bucket term, you can see that it's predominantly in the pie graph below. You can see it's predominantly the patent class C12N, which is microorganisms, enzymes. It's related to genetic engineering, followed by A61K, which are the preparations for medical, dental, or toilet purposes, and then C7K, which are the peptides. And then to further understand the countries that were where the patents were being filed, we saw that the top countries were China, the United States of America, Australia, Canada, and India. So then based on this analysis, we thought it would be a good idea to further look at the patent class C12N. And we can see that for the period in total from 2012 to 2023, we can see that there were almost 700,000 patents that were filed. And this, again, increased relatively linearly from about, in 2012, there were just over 39,000 patents filed to, in 2023, there was almost 82,000 patents being filed. So then to further, again, look at what was the breakdown within C12N, we looked at the subgroups. So you can see in the pie graph, you can see that it's predominantly C12N15, which is related to mutation or genetic engineering, plasmids, vectors, and the purification of those, followed by C12N5, which is related to undifferentiated human, animal, or plant cells and tissue culture, followed by C12N1, which is related to microorganisms. And then to then look again at the countries where the patents had been most filed, we could see that the top countries were China, the United States of America, Japan, Republic of Korea, and Canada. When looking further at the data and looking at the different patent classes available within the International Patent Classification System, we did notice there was this one class called G16B, which is related to bioinformatics, information and communication technologies, especially adapted for genetic or protein-related data processing in computational molecular biology. So because there was a similarity between some of the trends and issues, as well as we could see from some of the data that perhaps it could be a patent class to further explore, we did explore it. And what we could see is that for the period between 2012 to 2023, there were just over 32,000 patents that were filed. And this increased relatively, it started to pick up. So it started at about 246 patents being filed in 2012. And then around 2016, it seems to have shifted a bit and increased in the patents being filed to have in 2023, 7,401 patents being filed. And then again, to look at the further breakdown of the patents that made up the overall, we can see that the top groups were G16B20. So this was related to functional genomics or proteomics. Then followed by G1640. So this is biostatistics, bioinformatics related machine learning or data mining. And then G16B30, which is sequence analysis involving nucleotides or amino acids. And we can see that the top countries in this regard were China, the United States of America, Republic of Korea, Canada, and Japan. But just to also flag, there were some limitations to this literature review. It should be regarded as a preliminary analysis to complement the horizon scanning process. I think the major limitation that we had is time. So this was initially conducted in January 2024, and then following the peer review process redone in March and April 2024. Being that the search strings had to be adapted, the whole analyses had to be revamped. Another thing is that because of this, there is a bit of an asynchronous completion in relation to the work of the MATEG. Although the initial draft fed into the multidisciplinary AHTEG, the final draft would be feeding into the SABSTA meeting. In terms of the methodology, one thing to highlight is that this was conducted in Scopus, so other important publications and sources may have been potentially excluded, so those not registered within the Scopus database. As well as that, we were only able to conduct the analysis in English. So part of the methodology was using these Boolean search terms. So part of that comes that it's based on how the author self-described their work. So there is a potential that although we tried our best to update the search strings to capture as many relevant publications as possible, it is based on how the authors self-describe their work. There was a limited data cleaning. So generally in a traditional literature review that takes 18 months or, well, 8 to 18 months, there's a thorough review of the abstracts and titles. Given the time constraints, this wasn't unfortunately able to be done, but the strings were updated as much as possible to try to make them as relevant as possible to the trend and issue in synthetic biology. In addition, there were a few additional statistics and bibliometric analyses that would have been nice to have, but we were unable to complete based on the timing constraints. Further, for the trends and issues in synthetic biology, it's important to note, I alluded to this when I mentioned that that duplications were removed, that there were some overlaps between the trends and issues in synthetic biology. And then also to highlight that not all the trends and issues can be measured using a bibliometric approach like this. So there were certain trends and issues such as the transboundary movements, the inequity in the participation of developing countries, some of the cumulative effects. Some of those, because of the documentation may not be registered in the Scopus database, this approach might not be necessarily suited to those, but it was suited to some of the other trends and issues. And then just to also highlight that the current patent classification system doesn't differentiate between biotechnology and synthetic biology. So, for example, C12N is sort of a general patent class for all biotechnology. So although we explored it because it came up in our analysis as being making up the most relevant for our bucket term, it's just that there isn't a very strict definition in the patent system. So overall, you can see that there were linear increases in both publications and the patent filings in synthetic biology. You can see that in general, the synthetic biology-enabled production topics had relatively moderate growth. You could also see similar patterns observed between the publications as well as the patents. So you saw a lot of the patent classes and publications related to medical and therapeutic applications, but you also saw that the bioinformatics and artificial intelligence trends were also picked up strongly both in the publications as well as that emerging trend within the patent class G16B, as well as protein engineering. There was some difference between the countries contributing publications and the locations to where the patents were filed. You could see that both the United States of America and China were both leading the way in this regard, but you could still see that there were some slight differences that could exist. And then for the collaboration, there are opportunities that likely exist between the research groups. You could see that there's concentration in developed countries. However, when you could see from the maps that were presented in the collaboration analyses that developing countries and countries that haven't traditionally been involved in synthetic biology are also able to participate, perhaps through these research agreements. So with that, I would like to thank you for your attention, and I'll hand the floor back to Sanka. Thank you so much.
Thank you very much, Austin. And we would now like to open the floor for comments and questions. Yes, please.
Thank you very much for this very, very interesting 3 presentations that made me so alarmed. When I say that, number 1, synthetic biology progress is continuing at alarming stage. I keep asking myself, where is the ethics about it? This is the 3rd time today I mentioned ethical aspects in biodiversity, whether related to traditional knowledge, whether ABS or resource mobilization, and synthetic biology as well. Number 3, who is going to implement the proposed recommendations? We have 26 recommendations. Who are they? 5 countries, top countries, that they have developed all of this, where the rest of all countries as well. So from practical point of view, these recommendations will be incredible burden in developing countries to respond to all of these alarming stage of development. Because when I think just in about 10 years from now, how the artificial intelligence will deal with synthetic biology, where we will be. I do remember myself when I first get my PhD, maybe more than 40 years ago, and I look at it only 10 years later. I said, this is bullshit. If I have this data, I can make another thesis, whatever. So what I'm saying, literature review, tell us 2 things, either positive or negative impact. Big deal. We already know. But how can we respond to all of these alarming developments that faces the Earth? And that's why ethical aspects in biodiversity should be considered. In all our traditions, in all our religions, it is very clear. Stop damaging the environment or the Earth after it's been rehabilitated and restored by the God. Thank you.
Yes, thank you very much. And so let me just quickly reply to the second part of your comment. So, of course, we have a broad variety of potential recommendations in there, but those are primarily for the consideration by Substar. We are not recommending for parties or for specific parties to implement the full list of recommendations that come out of this process. So those are considerations for Substar itself. Regarding your remarks about ethics, I would personally tend to agree with you, although of course our mandate was to evaluate newest developments in SynBio vis-a-vis the 3 objectives of the convention. But that of course means that implicitly certain ethical considerations related to conservation, sustainable use, benefit sharing are already there. But other than that, I do see your point. Thank you. Over there, please.
Thanks a lot for the presentation and of course providing bibliometric information. And you talked about bioinformatics, machine learning and all that, and sometimes linkage between phenotypes and genotypes and the like, and how we can apply it in our systems. And especially I'm trying to link up with the self-limiting vaccines or so. So my question is like, for instance, with synthetic biology, probably, and with the computational ability that we have, we could even be having like new pathways. And what if some of these pathways can even take up like certain things like xenobiotics, and then you find out it's transmissible across probably our ecological ecosystems? How do we monitor? How do we control? And when something gets out of range, and with a self-propelling capacity, how to intervene sometimes. Those are some of the things that probably we need to reflect into. Thank you.
Yes, thank you very much. Personally, I'm just a political scientist, so since that is a very technical question, perhaps some of the other members of the group would like to pick up on that. Yes, please.
Thank you very much for the floor, and thank you for these good and informative presentations. Yeah, I have noticed that developing countries seem not to be considered in this process. Then I have a couple of questions. First one, can synthetic biology products be considered as LMOs according to Cartagena protocols? And second, can synthetic biology be considered as a new and emergent question? Thank you.
Thank you very much. And I believe the understanding is that most current SynBio products would classify as LMOs for the purposes of the Cartagena Protocol. I mean, regarding the question whether it's a new and emerging issue, that has not been resolved by the COP yet. So we are currently we're working with the operator. Latsi, please.
Just to respond to the question of new and emerging issues, you will recall during COP15, the decision was that we would delink the discussion on SynBio and new and emerging issues. So without prejudice to the process on new and emerging issues, COP took a decision to proceed with work on synthetic biology. And this is why we have this work happening. And you will note in decision 1513, COP also stressed that the fact that we have taken this decision on synthetic biology should not be seen as setting a precedent for topics coming onto the COP agenda in this manner going forward. So the discussions on synthetic biology now no longer have this question to answer about whether or not it is a new and emerging issue.
Thank you, Vatsi. Does that answer your question? Does that answer your question? Okay, then let's move on. Osama?
Yeah, just a comment to that. The issue of synthetic biology is discussed under the Convention on Biological Diversity and not under the Cartagena Protocol, basically. Because when we are talking about SynBio, we are talking about living modified organisms, part and product thereof. The part and the product thereof are not fully covered by the Cartagena Protocol. That's why we are dealing under the convention. That's why this AHTEG is under the convention of the CBD and not under the Cartagena Protocol. Thank you.
Thank you. Do you have further? Yes, back there.
Thank you, Mr. Co-Chair. I wanted to respond to the previous question before this one, which wasn't answered. So the simple answer is yes, you are right. Synthetic biology poses the complication that it can create products that are not necessarily already in the system. That could be peptides. So if you can synthetically create a gene sequence that you want, that means you can translate it into a protein that you want, and that protein can, you're right, integrate into existing systems. And the risk assessment AHTEG then has to figure out to help us, how do you then mitigate the risk that those proteins could cause in the natural systems. And in that is part of our recommendations actually in our document, that those are some of the issues that parties need to be considering as the work on synthetic biology goes on. How do you govern that? How do you even assess it? How do you know where the products of synthetic biology are going to end up? And that needs real assessment, but I think each case you deal with it case by case. It depends on what the product is that you're actually looking at, what the product is that you're hoping to introduce into the environment. At this stage, because there hasn't actually been any product that cannot be assimilated and gotten rid of, the scary answer is we don't know. So you don't know where the protein you're introducing is going to end up. How is it going to affect existing biological systems? We don't know. That's where risk assessment comes in. You have to then try and put measures in place, governance in place to deal with that, to say, how will we follow up? How will we monitor whatever product we are introducing? I hope that helps to answer the question.
Okay, thank you, Natasha. And over there, please.
Thank you for the floor. My name is Nancy Marangu. Thank you for the wonderful presentation. My question is, what tools might be in place to monitor and evaluate the use of the geospatial technologies that you've spoken about? And what metrics, again, will you use to determine and decide which technologies will be harnessed when? This is with the appreciation that different countries have different technological infrastructures. Thank you.
Yes, so the monitoring aspects is something that we have not looked at in depth yet. So of course, the overall terms of reference of the group were for assessment, horizon scanning, and monitoring, but we have focused on the horizon scanning and assessment parts, and monitoring, that is something that should be a priority for potential future iterations of this group. Do we have other comments or questions? Over there, please. Eva?
Thank you. I don't have a question, but just a comment on the monitoring aspect, which I think was something that we discussed quite a lot in the meeting. So yeah, I think it's also a big concern, particularly with one of the topics like self-spreading vaccines, where monitoring seems to be, it raises challenges for the current process we have for monitoring. So that was something that we did raise in the meeting. For example, self-spreading vaccines, they're targeting hard to reach wildlife. So how you would monitor changes in this self-spreading viruses and these issues are really challenges that we have to current risk assessment protocols. So yeah, I think monitoring, I agree, is a very important topic. And we that do challenge current processes, and we don't have the solutions for that at the moment. And that is something that came up quite prominently in the meeting. Thank you.
Thank you. Do we have other comments or questions?
Thank you very much for your discussion, comments, and questions in this session of our workshop. I think we are coming to the end of this workshop, but before we leave, I would like to remind you how long was the day and to ask chairs or co-chairs of these three AHTEGs just to summarize very shortly what you have had today, what you had the chance to hear today. I would like to ask co-chairs of AHTEG on indicators to just to summarize this part of workshop today. Thank you.
Thank you very much indeed, and thank you everybody for the conversations that we had this morning. The monitoring framework was adopted at COP15, including headline, binary, component and complementary indicators. And the AHTEG has undertaken a huge amount of work to facilitate parties making that monitoring framework operational. We've heard quite a lot this morning about a number of different subjects around that, and there are some challenges. There are clearly some challenges, but I think it's now necessary to endorse the work that's been done to try to make sure that the monitoring framework is complete, or as complete as we can do. Indicators are always going to be a subject that is evolving. I think it's likely that we will need to continue to consider how better to monitor the coming Montreal Global Biodiversity Framework going forwards. But that is not an excuse to say, no, it's not ready for implementation. It is ready for implementation. It's ready for implementation, 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 because we need that in order to be able to understand better the world in which we're living, and in order to be able to understand the progress that's being made towards the implementation of the ambitious coming Montreal Global Biodiversity Framework. And I'll finish by making a plea for simplicity in the conversations around the binary indicators that we may be having later in this week. It's very tempting to make the questions more complicated. But actually, remember your colleagues who are going to have to implement those questions, who are going to have to answer those questions. There is a virtue in simplicity. And remember, we may not be able to measure everything, but we have made huge progress compared to where we were with 2010, and we will continue to make further progress as we move towards 2030. So I commend the work of the AHTEG to parties and hope that you will support it. Thank you.
Thank you very much. For the moment, I don't see chair of AHTEG on risk assessment in the room. Then I will ask co-chairs of AHTEG on synthetic biology just to say a few more words about this work.
Okay, thank you. Then for final considerations, I believe that all of us are quite satisfied with the outcomes and that we managed to navigate a very complex process with a broad diversity of views in a very short period of time and in a manner that was constructive and collegial. And I believe that considering what is at stake for the objectives of the convention in regards to the very fast-moving field of synthetic biology, I believe we did a decent job, and none of that, of course, would have been possible without the invaluable support of the Secretariat. So at this point also, once more, thank you very much, Austin, Batsy, Melissa, and the rest of the team. Thank you.
Thank you. And I think we will close our workshop now because I think we are at the end. For the AHTEG on risk assessment, we, of course, realize that outcomes are really very, very important for the future and, of course, now for the Substat 26 conference, as all three are. I believe that we have a productive and informative workshop. And my colleagues in Bureau and I thank you for sharing your views and perspectives and for your constructive engagement. Many thanks to those who joined us online from their respective homes. Thank you for your participation and interest. I heartily thank all the co-chairs for their presentations today and for their dedication and valuable contributions to these important processes. I sincerely hope that today's information workshop was helpful to all participants in describing the excellent work of three expert groups and that it has helped to inform your upcoming deliberations at Substat 26. I'm really very glad you had the chance for direct exchange with experts today. I thank you all for your participation and discussions. Now I wish to ask Secretariat if there is any announcement before we close. No new announcements from Secretariat, then I thank you all.